Hello friends, welcome to BMH Learning. In this video, we are going to talk about isotachophoresis. The technique is based on the principle of moving boundary electrophoresis. On the basis of equipment being used, separation can be done either in a horizontal or vertical way. Now let's split the term isotachophoresis. Iso means same. Tacho signify speed and phoresis means travel. When we combine these, it refers to the fact that all the ions being separated are actually traveling at the same speed. The ionic components of sample mixture are separated in such a way that they are stacked into discrete zones. The separation rate depends on the order of their mobilities. Now, what does this mean? It means we are using a discontinuous electrolyte system. So, we are using two electrolytes. Le stands for leading electrolyte, that is the one which is ahead in the race, whereas Te stands for trailing electrolyte. We can say the one which is behind in the race. Now the thing is that the sample solution is applied at the interface of Le and Te. Now, how does the separation of mixture of ions occur? So you need to know that a leading anion has higher mobility than sample ion. Likewise, a trailing ion has lower mobility than the sample ion. So that simply means the analytes of interest, or in other words, the ionic components we want to separate, have intermediate ionic mobility. So when the current is switched on and electric field is established, the ions in the Le will migrate towards appropriate electrode. Talking about the sample ions, they would follow the migration speed with respect to their electrophoretic mobilities. As we know about the ions in Te, they will follow the sample ions. After some time, a moment would come when there will be an equilibrium in all ionic species we are talking about. So at this time, all the ions will move with the same speed in discrete zones, depending upon the order of their mobilities. Few important points. In cases where the sample ions have similar electrophoretic mobilities, in this situation, synthetic ampholytes called spacer ions can be included to enhance the resolution. The important feature of these spacer ions is that they have mobilities intermediate to that of sample ions. Thus, intermediate mobilities of the spacer ions helps in separation of sample ions by taking up positions between the sample ions. The spacer ions are similar to the ampholytes which are employed in isoelectric focusing. Applications of isotachophoresis. The method is used to separate charged substances that range from inorganic to organic acids to proteins and nucleic acids. This technique is one of the most widely used in research laboratories. It's very useful in industries for pollution control, as it's used for the detection of detergents and also the inorganic ions in effluent water. In case of food, brewing, and pharmaceutical industries, it's used as a quality control method. There is an added advantage of this technique. Very small amount of sample can be separated. This was all. Thanks for watching.